saying a lovely afternoon, good morning, as well as evening to those that have been able to join us on today's Echo. I'd like to warmly welcome you to today's session. My name is Zoran Muhimba. I'm a consultant pathologist here at the University Teaching Hospital, and I'll be the facilitator for today. As we all know, AMR poses a serious global threat to the animal, human, as well as environmental health sectors. And you will agree with me that the sub-Saharan region has bore the heaviest burden of these resistant bacterial infections. And it is from this background that today's session will be looking at one of the 5 pillars for the implementation of integrated antimicrobial stewardship activities that have been recommended by the WHO. And this is looking at the role of IPC as well as WASH in the control of AMR. Now, to help us discuss this exciting topic, I have here with me Dr. Chileshe Lukwesa, who is one of the key players as well as mentors in the AMR space in Zambia. Now, Dr. Lukwesa is a consultant clinical microbiologist that is working at Lusaka District Health Office under the Ministry of Health. She's also the national focal point person for AMR for the human health sector, and she's a member of the technical working group on AMR in infection prevention. The other presenters that will be presenting a bit later on the case presentation, we have Dr. John Tembo, who is a scientific coordinator under Herpes Zambia. Then we also have Dr. Lindsay Elton, who is a postdoctoral researcher from the Center for Clinical Microbiology at the University College in London. Our panel of experts includes Professor James Mwansa, who has a PhD in medical microbiology from Manchester University, UK. He's also an associate professor of medical microbiology, as well as director of research and postgraduate studies at the Lusaka Apex Medical University. We also have Mr. Innocent Hamuganyu, who is the IPC coordinator for WASH and healthcare, and he's based at the Ministry of Health. Among the panel, we also have Mrs. Alfonsina, who is at the University Teaching Hospital Adult Hospital and is an environmental health officer. Our panel is concluded by Mrs. Musonda Mandona, who is a biosafety and biosecurity advisor at the Ministry of Health. So a few reminders on this session today. Please ensure that all the gadgets that you're using are muted, and even as we'll be opening up for plenary after the presentations, kindly introduce yourself before you are able to ask the question. And please feel free to be able to use the chat room for any questions that you may have. We'll be able to look at these once the presentation ends. So with that, I'm going to invite Dr. Chileshe Lukwesa to take us through the first presentation. Doc, over to you.
Thank you so much, Facilitator. So today's topic is the role of infection prevention and WASH in antimicrobial resistance control. So this is a topic we continue to look at a series of topics which are informing us on the preservation and effectiveness of antimicrobials. So there will be other related topics which are related to preserving the effectiveness of antimicrobials. So today we look at the infection prevention and WASH in antimicrobial resistance control. Next slide. Okay, so what are our learning objectives for this session? So we wish to understand the role of IPC, infection prevention and control, and WASH in preventing and controlling antimicrobial resistance. We also look forward to understand the principles and strategies used for IPC and WASH, and also understand the consequences of poor infection prevention and control practices of WASH. So we'll start by defining the terms we've just been mentioning. I think it's worth understanding the terms so that we understand the content of the presentation quite well. So when we talk of infection prevention and control, according to WHO, infection prevention and control is a scientific approach and practical solution designed to prevent harm caused by infections to patients and also to healthcare workers. Healthcare-associated infections, HAIs, we'll talk about HAIs, and these are infections people get while they are receiving care for another condition while being attended to in a healthcare facility. And then WASH, these are acronyms, which stands for water, sanitation, and hygiene. So we'll go to our first poll.
So we have our first poll, which is being launched right now. So the poll is looking at, ask the question on infection prevention and control and WASH as being one of the strategic interventions in the multi-sector Zambian National Action Plan, or the Zambian NAP, on antimicrobial resistance to control the emergence or increase in antimicrobial resistance. So we have three options. The first being true, option B being false, and option C being not sure. So we'll give everyone a minute to be able to give us, to be able to fill in their poll. So please feel free to put in your option. We still have a few seconds to go. Okay, so I can see that all 24 of us have been able to fill in our answers. So we have those that are saying the answer is true. We have about 92%, which is representing 22 of the 24 who are in this session. And then we have one that has said false, and one has mentioned that they're not sure. Thank you so much. So we'll continue, we'll be able to come back to the answers to these polls at the end of the presentation. Please continue the purpose.
Okay, thank you. Next slide. Okay, so that is our the image of our multi-sector national action plan on antimicrobial resistance. And this national action plan has got 5 strategic interventions to combat antimicrobial resistance. So there's an issue of awareness in Objective 1, raising awareness. Objective 2, producing evidence through surveillance. Objective 4 is optimizing drug use. And Objective 5 is investing in new technologies. And we can see that Objective 3, as a strategy to combat antimicrobial resistance, is to reduce the incidence of infection through effective sanitation, hygiene, and infection prevention measures, and biosecurity. So let's look at the current situation on healthcare facilities in terms of WASH, IPC, and AMR. In low and middle income countries, this data is taken from WHO and UNICEF. And under WASH, we should note that 38% of healthcare facilities do not have any water source, okay? 19% do not have improved toilets. This is in healthcare facilities. 35% do not have water and soap or alcohol-based hand rub for hand washing. Up to 90% of healthcare workers do not adhere to recommended hand hygiene practices. In terms of IPC, in Africa, up to 20% of women get a wound infection after a cesarean section. Hospital-born babies in low-income settings are at a higher risk of being affected by neonatal sepsis, with infection rates 3 to 20 times higher than in high-income settings. And then on average, 15% of patients who acquire at least one infection while they are being cared for in our hospital settings. Antimicrobial resistance, what is the status? Prophylactic use of antibiotics is standard in over 80% of maternity units in several countries. That's a high use of antibiotics. And then our patients with resistant Staphylococcus aureus, this is a common pathogen found in hospital setting, acquired from hospitals. The resistant strains of Staphylococcus aureus, 50% will be more likely to die from the resistant strains than from a non-resistant infection due to Staph aureus. And each year, hundreds of millions of cases of diarrhea are treated with antibiotics. Universal access to WASH could reduce this by 60%. So how is infection acquired? What are the players in acquiring infection, whether outside the hospital setting or within the hospital setting? So we've got the pathogens which will cause the infection. If you look at that chain, there should be a reservoir where the pathogen is allowed to multiply and survive. We need to have a mode of escape from that reservoir. And we need to have a means of transmission of that organism from the reservoir to a susceptible host. And within that susceptible host, there should be a mode of entry. So these are the players in the chain of infection in our healthcare setting. Next slide.
So we have a second poll question, which is asking the question, at which stage is it possible to disrupt the transmission of infection? So option A, at the reservoir. Option B, at the reservoir, portal of entry, and portal of exit. Option C, at the portal of entry, at transmission stage, and at portal of exit. Option D, at the reservoir, portal of entry, and portal of exit. And option E, at all stages of the chain of infection. So we have a poll question. Again, we'll give it about a minute for everyone to be able to put in their options. Please kindly note that let's choose the option and not put the answers in the chat room. Let's click where we think the answer could be, and then we'll be able to look at it at the end of the presentation. Thank you. Okay, so again, we'll end the poll. We'll be ending the poll soon, just a few minutes. Okay, I can see that all 33 of us have been able to input our answers. So I'll end the poll now and able to share the results. So we can see that for option A, 3 of us have given option A as an answer, which is 9%. No one has given option B, and then we have 3 as well that have chosen option C, which is at the portal of entry, transmission stage, as well as at the portal of exit. Then we have 1 that has chosen D, which is at the reservoir, port of entry and port of exit. And then we have 28, representing 80%, that have chosen at all stages of the chain of infection. So thank you for that. We'll continue with the presentation at this point.
Thank you so much. Just to introduce the concept of healthcare-associated infections, we define that. So healthcare-associated infection, sometimes referred to as nosocomial infections or hospital infection. So these are infections which occur in a patient during the process of care in a hospital or other healthcare facility. Other healthcare facility may be a hospice or any healthcare facility where you are providing healthcare. And this is an infection which was not present at the time of admission. So these are infections where somebody comes in with a different infection and acquires another infection, or different condition, but acquires an infection in the hospital. So within the hospital, how are these infections transmitted? They can be direct transmission or indirect transmission. So direct transmission can be direct contact skin to skin, or it can be droplet spread, where somebody is sneezing or coughing and you get the infection through that. Or it can be indirect transmission through airborne transmission, where particles are suspended in the air and somebody inhales those particles containing infectious agents. Or the infection can be transmitted indirectly by vehicles. Vehicles may be food, water, blood products, or other inanimate objects, which we refer to as fomites, which include beddings, surgical instruments, and so on. And then you can also have indirect transmission through vectors. For example, by bite of a mosquito, somebody gets malaria, and the vector is a mosquito. Okay, and other vectors can also apply. So this is an example of sources of infection in a healthcare setting. These are real results coming from a sampling from an operating theater within one of our facilities. And we can see the sampled sites, the suction tubes, the scrub sinks, operating bed, oxygen tubes, and so on. And we see that some of these do harbor some pathogens or microorganisms. If we looked at those organisms, most of those are organisms which are found in the gut, in the GIT, and they'll find themselves in our tubes, in our sinks, and so on, if infection prevention is not implemented. So these are real results. So, however, there are opportunities to break the chain of infection. Transmission may be interrupted when the infectious agent is eliminated, inactivated, or cannot exit the reservoir. We can break the chain of transmission at the portals of exit. The portals of exit can be contained through safe infection control practices. The transmission between objects or people does not occur due to barriers and safe infection control practices. The portals of entry can be protected, and other persons receiving healthcare who are not susceptible. So this is the same chain, and we can, this is just highlighting what we can do to prevent transmission at each stage. For example, at the infectious agent stage, the rapid detection and identification, healthcare workers will react and deal with that infection which has been identified. The reservoirs, which can be people or healthcare workers, the environment, infection can be reduced, or organisms can be reduced in the environment by sterilizing and disinfection of the environment, or the healthcare workers can be treated if they have an infection and so on. Environmental sanitation, important there as a reservoir. At portals of exit, trash and waste disposal of excretions, skin and droplets. So those can be taken care of. Infection prevention measures can be taken, including proper attire, hand washing, and control of excretions and secretions. The modes of transmission we've looked at, we can disrupt that, for example, by proper food handling, by isolating patients, and by hand washing. Portals of entry into the susceptible host, we can prevent infection from getting to the susceptible host by ensuring aseptic techniques, proper wound care, catheter care, and other preventive measures. And then we should also be able to recognize the susceptible host. For example, the immunosuppressed patients, the diabetic patients. We need to recognize these high-risk patients and take intervention or take preventive measures. Next slide.
Okay, so again, we're going to have another poll question. This is asking us, whose responsibility is it to champion infection prevention and control in healthcare settings? So we have several options here. We have option A, he's saying infectious disease specialist or trained physician. Option B, pharmacists. Option C, infection prevention nurse. D, unit nurse champion. E, environmental health practitioner. And F, microbiology laboratory staff. And then G, all of the above. So again, we're going to give it about a minute so that you can be able to choose your options, and then we'll be able to share the results. So at the moment, we have 58% of us that have selected our options. We still have a few seconds. It would be wonderful to have everyone choose their option. Remember, this is just for learning purposes. We are now at 60% of the participants. We'll give it a few more seconds. Okay, so I think 60%, we still have a few more. Okay, so I think we'll be closing the polls now. So I'm going to share the results. Okay, so we have one person out of the 48 that selected their options, who thought that an infectious disease specialist or trained physician is the one that is responsible. Then we have 2 that are saying environmental health practitioner should be the one responsible. And then we have 45, which is representing 94% that are saying all of the above. Okay, thank you for that. So we'll continue with the presentation.
Okay, so thank you so much. So, infection prevention champion can come from anybody. So it is a multidisciplinary effort. No one person can work on their own and ensure that there's infection prevention. It's a teamwork. It's a disciplinary team which is involved in infection prevention. The infectious disease specialist provides medical expertise, to infections and their prevention, a trained pharmacist who offer insight in the prudent use of medications or antibiotics. Infection prevention nurse will focus on implementing infection control practices and protocols. The unit nurse champion will advocate for infection prevention within specific units. The environmental health practitioner will lead us in environmental health hygiene, and the microbiology staff are important in the early detection, surveillance, and early warning of any outbreak and investigation. So what are the principles or strategies which can be used in infection prevention? We've seen that circle there. The outer circle is the largest. A very important intervention or strategy is hand hygiene. Hand hygiene before touching a patient, before any procedure, after touching a patient, after touching patient surroundings, and after body fluid exposure. At all these points, we need to ensure that there's hand washing. So the other strategies are environmental hygiene, screening and cohorting patients, surveillance, prudent use of antimicrobials, antimicrobial stewardship, and following guidelines, as well as a safety culture. And what are the strategies for water, sanitation, and hygiene? In our healthcare facilities, we need to ensure that the sufficient water is available at all times from an improved source located on the premises. There's access to hand hygiene facilities, clean hygiene conditions, improved sanitation facilities located on functional premises. We need a well-lit areas and adequate ventilation and drainage system, adequate power for sterilization and incineration of medical devices, adequate supply of PPEs, separation of waste and safe disposal, and also management of sewer and fecal waste. So if all this is not done properly, if IPC and WASH are not implemented adequately, what are the consequences? So we'll see that there will be increased risk of healthcare-associated infections, increased risk of the spread of healthcare-associated infections, increased burden of expensive, hard-to-treat, and life-threatening resistance infections, and decrease in patient confidence in healthcare. And maybe the previous slide. And all this will lead to over-reliance on preventive use of antibiotics, that's overuse of antibiotics, high healthcare costs and poor health outcomes, increased use of antibiotics to treat preventable infections, and of course, increased antibiotic resistance. And the opposite is true. Effective infection and control and WASH measures implementation will lead will reduce the spread of all types of infections, including resistant ones. The WASH and IPC will also reduce the overall antibiotic demand. The use, misuse, and waste will be reduced and reduce the opportunity for resistant strains to emerge. So there will also be fewer resistant infections, and this saves money, and better WASH and IPC will lead to reduced antimicrobial resistance overall, as we will not be able to use antibiotics which and the antimicrobial resistance. So infection prevention in healthcare facilities is necessary to minimize antibiotic use. And we've talked about the measures. Next slide. So in summary, effective infection prevention and control and water, sanitation, and hygiene WASH stops the spread of antibiotic-resistant organisms. IPC and WASH in healthcare protects patients, the healthcare worker, from avoidable infections. Every infection prevented is an antibiotic treatment prevented. And we've learned that all categories of healthcare workers play a role in infection prevention towards control of antibiotic resistance. These are the references. And we can go to the repeat pause.
So thank you so much, Dr. Lukwesa, for that wonderful presentation. Quite insightful, and we hope that you've been able to pick out one or two things from the presentation. We'll now go ahead and do a repeat poll of the poll one, which we looked at first. So we'll launch it again, and then be able to share the results. Okay, so poll one reads, infection prevention and control and WASH is one of the strategic interventions in the Zambian National Action Plan to control the emergence or increase in antimicrobial resistance. So again, we'll give it, I think, 30 seconds in the interest of time. Please feel free to put in your option. At the same time, please try to ensure that you click on the poll which has been shared and not in the chat group. Thank you. We still have a few seconds. Currently, only 18 of us have been able to respond to the poll. Okay, we now move to second poll, which is asking the question. Sorry about that. That's a different poll. So we'll be changing the poll two soon. Okay, there we go. So the second poll question is asking us, at what stage is it possible to disrupt the transmission of infection? So please feel free to indicate your option and ensure that this is on the poll provided and not in the chat group. Thank you. Okay, we have 37% who have been able to answer this second poll question. We're at 50% now. Okay, so we now move to the next. So the third poll is asking the question, whose responsibility is it to champion infection prevention and control in healthcare settings? Again, we have several options there, starting from infectious disease specialist or trained physician, pharmacist, infection prevention nurse, unit nurse champion, environmental health practitioner, microbiology laboratory staff, and all of the above. We have a few more seconds to go. We have 50% have been able to participate in this poll. Please feel free to select your option. Okay, so we'll end the poll here. And now we'll go straight ahead and look at what are the answers to the poll questions. So we have poll 1, which was asking us on IPC as well as WASH, as being one of the strategic interventions in the Zambia National Action Plan to control the emergence or increase in antimicrobial resistance. And you agree with me that Dr. Lukwesa did highlight that we have 5 strategic interventions, and of which intervention number 3 speaks towards infection prevention and control. And so the answer there is A. Then we move on to poll 2, which was asking us the question, at which stage is it possible to disrupt the transmission of infection? And again here, she did highlight in the presentation that all the stages, we can actually be able to implement, put in interventions to actually prevent the transmission or to disrupt the transmission of infection. So the answer there is E. And then the last poll, which was asking us about the responsibility as to who it falls to. Again here, she was able to highlight what role each one plays, and that all those that are involved in healthcare have a responsibility to play when it comes to infection prevention and WASH intervention. So our answer there is D. So again, to just appreciate Dr. Lukwesa for the wonderful presentation. We'll now move on for the next few minutes to look at any questions that may be arising from this presentation. Currently, I'm looking at the chat room, and there are no questions at the moment. I think just one concern that a very interesting presentation. Again, this is from ITEL S662L, who is asking if the presentation could be shared on the group. I think this can be done. We'll be able to share the presentation. Okay, then Kapula Chifunda says, well presented. We have quite the fight ahead of us if basics such as safe running water are not accessible to everyone in health facilities. Yeah, this is really true and quite concerning. I think we're able to see those numbers. Okay, so are there any questions? I've not seen any hands being raised. Okay, if there are no questions, we will now proceed to the second part of our ECHO session, which is looking at patient case presentation. And again, I must mention that we have 2 presenters for this one. Okay, we have Dr. John Tembo, who is a scientific coordinator and for Herpes Zambia, and then we have Lindsay Elton, who is a postdoctoral researcher from the Center for Clinical Microbiology at the University College in London, UK. So please go ahead.
Hello, everybody. My name is Lindsay, and I think I am starting. So if you'd like to put up the first slide, please. I'm not sure who has the slides.
Noted, just give us a minute. We'll be able to put up the slides soon.
That's okay. I can kind of explain the background then while you're doing that. So yes, this is just a brief talk about how we've been developing a set of genomic tools to help understand and identify transmission of pathogens within hospital settings. So what we've done is developed sequencing tools, so whole genome sequencing and also metagenomics, to look at patient samples and also from swabs within the hospital. So we've done systematic swabbing in the UK, going around and sampling parts of the hospital, so things like door handles and toilets, and taking wastewater samples from sink U-bends and things like that. We've created, we did whole genome sequencing on the patient isolates, whole genome sequencing on some of the isolates we've extracted from those environmental samples, and then also did direct metagenomic sampling. So that's kind of everything that exists within that sample to look at transmission. And ultimately what we want to do is try and identify how we can use these tools to help inform good or better infection prevention control practices, because if we can find reservoirs of particular organisms that are of concern in one ward or another ward, then we can start to target IPC practices. So that's what we've been developing here in the UK. And then, John, I don't know if you want to, while we're waiting for the slides, to discuss what we did in our COVID project. Oh, never mind. I'll start now. So yes, I think I probably explained a lot of this already. So what we are planning to do is build this pipeline to identify both pathogens, so multi-drug resistant pathogens or MDROs, and antimicrobial resistance transmission on wards. So what we sort of ideally want to find is, where are these infections coming from? Are they community acquired? Are they sort of environmentally acquired? Or are they being acquired from sort of transmission via healthcare worker hands and things like that? So as I say, we use whole genome sequencing and metagenomics. And on the right, you can see a flow cell. So we've been using Oxford nanopore technology. That's one platform for whole genome and metagenomic sequencing. You may have heard of it. If not, the reason we have chosen to use it, well, there are many. It's got a relatively cheap initial setup. So it's about $1,000 for the starter pack. And it has and also a very small laboratory footprint. So it's about the size of your hand. It's kind of like a dongle that you can attach to either a computer or to a laptop. So it's small, it's cheap, it's mobile. And so it's very, very useful for sequencing in sort of smaller laboratory setups. Oxford Nanopore Technologies uses long read sequencing. So Illumina uses pieces of DNA that are about 200 to sort of 400 or 500 base pairs long, so quite short, whereas Nanopore can create DNA sequences to read of about, well, up to 10 or 100 mega bases long. So the average size in this one that we were working with was something about 16,000 base pairs. So it's much, much longer. And that makes it much easier to piece together the puzzle that is sort of a whole genome. So if you think of a genome as like a puzzle, it's much easier to put a puzzle together if you've got 4 massive pieces than it is if you've got 100 little tiny pieces. So long read sequencing can be really useful for that, especially when you're doing metagenomic sampling, because with metagenomics, you've got all of the different species that are floating around in your water sample, and you've got to piece all of those together into the right sort of species. So long read sequencing is really, really helpful for that. It also has a relatively straightforward laboratory sort of side of things. So doing the extraction, growing up the bugs, doing the extraction, and then preparing the sequencing library and then running it. It's much, much quicker than Illumina tends to be. So down at the bottom, you can see the kind of flow chart of what we were trying to achieve by developing this pipeline. So we wanted to get the right extraction of the DNA, trying to combine speed versus the amount of DNA you need to input and the quality. Then with the library preparation, how much DNA do you need to put in versus how much information are you getting out of it? Then base calling, which is converting the sequencing data from raw information to the A's, T's, G's, and C's. And with this is a computer-based thing. You want to kind of balance accuracy. So obviously you want it to be as accurate as possible, but also speed and computing power, because the more accurate these programs are, the longer and the more computing power it takes. And sometimes doing really high accurate base calling can take a GPU, so a very, very powerful computer, some weeks to finish base calling. And that's obviously, if you're putting it into a diagnostic pipeline, then that's not very practical. And then finally, at the end is the analysis of that data. So again, accuracy, you want to make sure what you put in is what you're getting out in terms of information. But it also needs to be relatively easy to use, because there's no point creating a tool that is only sort of available for the 1 or 2% who are capable of doing really sort of command line based bioinformatics. We want it to be available, for everybody to be able to use. Laboratory personnel, clinical personnel, we wanted it to be as useful as possible. So just at the bottom is what we ended up kind of going with. So for extraction, for gram negatives especially, and gram positives in general, as long as you have validated your extraction kit, any of those off-the-shelf ones generally tend to work quite well. In terms of library preparation, the rapid barcoding kits came out best because they're quick, they're easy to use, and you can multiplex, so you can put multiple samples on one run, making it much cheaper. In terms of base calling, that kind of depends on what it is you've got available in your laboratory. So if you're doing high accuracy base calling, if you have a fast enough computer, then that's definitely the best. But if you don't have a fast computer and you need the results quickly, then fast base calling, as long as it's been validated, does work perfectly well as well. For analysis, there are loads of different tools that you can use. In terms of simplicity, the Center for Genomic Epidemiology is a website that has lots of really useful online tools that work within the browser. You don't need to have line command understanding. You can just drop your information into it and it will give you the output. So that was kind of the tool that we have developed. And then if you go to the next slide, please, I can explain what we found in our hospital. Thank you. So in the hospital that I work in, so this is a Northwest London tertiary hospital. In 2022, we had an outbreak of OXA48 mediated carbapenem resistant organisms in a number of patients across a number of different wards. Based on the patient metadata, so their bed movements, the times they were there, where exactly they were in the hospital, the clinicians realized that there's possible transmission going on here, that these aren't just sort of individualized cases. So what we did is use this pipeline. We took samples from patients. We took samples from one of the main offending wards, and we compared and combined these. And so we looked at a number of different species, but in the interest of time, I'm just going to show you the information we found for E. coli. This is a phylogenetic tree of all of the whole genome sequenced isolates. So patients and environmental isolates. So anything that says EI is an environmental one, and anything that says patient is patient. Hopefully you can see down at the bottom in the big blue box, you can see that there's a cluster of E. coli's that are almost identical. They all have the same strain, and you can see that on the right-hand side, they all have the same resistance sort of patterns. So we think because these are all patients, it looks like there might be patient-to-patient transmission. And funnily enough, they were all actually, apart from one of these patients, housed on different wards while they were in the hospital. But based on all of the other metadata, they were actually overlapping in all of these other wards. And what we found is actually utilizing that data is very complicated because everybody is moving around and trying to trace beds is very difficult. Something else that's quite interesting here is the one just above that. So the VREC0864, you can see this tiny cluster of two is an environmental isolate and a patient isolate. And they, again, are very closely related. They're the same strain, although they do have different resistance patterns. But these, we think, is an example of either patient to environment or environment to patient transmission. This patient and this environmental sample, which came from the waste trap, so the U-bend of the sink in the shared use pantry, they were pretty much identical. So as I say, either that patient who may well have been colonized and was unaware perhaps that they had this organism within them. So it could be the fecal oral transmission. Perhaps they had a drink and they tipped it down the sink maybe, or whether they got it from the pantry and then they were colonized with it themselves. At this point, we're not quite sure, but we can see that there is potential for this transmission based on our data. So it's potentially then, if we're looking at IPC, we might want to think, well, maybe we need to remove that U-bend and replace it in the pantry, because that might be a reservoir where patients are then from the environment getting infected or colonized from. And in terms of the patient-to-patient transmission, maybe there needs to be some IPC intervention looking at perhaps healthcare workers, as they may well be going between all of these patients and treating them. Perhaps we need to look at the use of gloves or the use of hand washing. So whilst this doesn't give us solutions as such, it gives us an insight into how solutions might be implemented. So if you just go to the last slide on my part. This again is just the E. coli, but it was a similar sort of representation across the different CRE isolates that we got. We found 9 clinical E. coli isolates and 11 environmental isolates. And in almost all, apart from one of the clinical, so in the UK, we use a rapid, sorry, can you just go back to the, the other slide? Could you go back to the last slide, please? Sorry, I'm just going to finalize the table.
That slide.
Just the one before, if that's possible.
Slide number 3.
Other way. There we go. Excellent. Yes, so I'll just finish off by saying in the UK, to rapidly identify whether there is carbapenemase resistance in patient samples, we use something called the CARBA5 panel, which is it's a genomic-based lateral flow test. And there's a sort of panel of oxygenes that are indicated on that. So whilst none of the patient or environmental isolates had BLA OXA48, which is kind of like the flagship oxygene, almost all of the patient isolates did have an oxygene that was indicative of carbapenemase resistance. Although, as I say, the environmental isolates didn't. There's various different reasons for this. So the uptake or removal of plasmids or the AMR genes when you go between the environment and patients. So in patients, they are generally treated with antibiotics. So it's beneficial to have these plasmids with resistance genes on. If you get a new pathogen that doesn't have these genes, it might be that the commensal organisms within the guts, for instance, will share those resistance genes. In the environment, it might be that they lose them because for antibiotics, you're far less likely as a bacteria to come into contact with antibiotics. But what we did also find is that these environmental isolates had far more genes that conferred resistance to disinfectants and environmental, decontaminants. So things like chlorine and stuff like that. So yes, it's kind of, you adapt to your environment, I think, in terms of what genes you have and also what you switch on and off. So again, understanding what genes are present helps to kind of identify what you could potentially use as IPC. If all of the organisms have resistance to a certain disinfectant that you're using, then you might want to change to a different one, for instance. So I will stop there, and thank you very much for inviting me to talk.
Thank you so much. We'll move to the next part of the presentation. So we'll have about 5 minutes for this next part. Thank you.
Okay, all right, I'll make it quick. So following on from the work that Lindsay was doing, we decided to use the pipeline within our context, within the hospital in Zambia, as well as we had another site in Sudan. But what I'll be talking about will be the Zambian results, as they'll be, I'm guessing, more interesting to the group. So we use the same Oxford nanopore sequencing techniques and the same pipeline. And our aim was to see during the COVID outbreak, we had patients who were being treated on the COVID wards and, the other regular wards on our internal medicine wards. So we wanted to see if there was, due to the high use of antimicrobials at that time, in trying to treat co-infections on the COVID wards, we wanted to test and see if hospital-acquired infections, were showing different sort of patterns of resistance or antimicrobial resistance on the COVID wards as compared to the non-COVID wards. So if we go to the next slide. Yeah, so basic main findings were that on the COVID wards, we had a higher number of MDR isolates as compared to the non-COVID wards. This was both in Zambia and Sudan. We also noted that in Zambia, the number of genes per isolate that were conferring beta-lactamase resistance was also higher. This was in the genotypic, when we look down further in the genotypic analysis. So, following on from that, we've noticed that obviously there's a problem with antimicrobial resistance on our wards. And, we're trying to look at which wards we could have more impact in looking at antimicrobial resistance using the same sort of pipeline that Lindsay has suggested, and that we've used previously in this study, which was published. So just the next slide, which will be the last one. So we are now hence planning on using this whole genome sequencing and metagenomic sequencing on the neonatal intensive care unit. The aim is to analyze patients, samples from patients with sepsis, analyze environmental samples, basically any patient with a bacterial infection, also analyze environmental samples as well as samples from the healthcare workers to see if we can kind of piece together the transmission pattern where we're seeing AMR being introduced into our wards. Yes, so I will end there.
All right, thank you so much for that wonderful presentation. So as a summary of what the case presentation was on, and again, Lindsay and John, please feel free to correct me if there's any aspect you want me to highlight. So to start with, the case presentation was mostly focusing on how we can use metagenomic sequencing in terms of aiding us to see where we are having these resistant pathogens arising from. I think we were able to pick out that they did a number of isolates. I think she did highlight that from the isolates which they picked, they got 9 from the clinic, which were clinical isolates, and they got 11 from the environment. And they found that after doing metagenomic sequencing on these isolates, they were able to pick that all 9 of the clinical isolates actually had changes within them that were more speaking towards resistance, and therefore they were able to come to the conclusion that the transmission in this case was not from the environment to the patients, but rather was between patients. And then later on, John did look at one study they did, I think, during the COVID time, where they were able to find, again, they were looking at whether these are coming from the environment, as well as whether they're between patients. So as a way of summary, we'll probably say that we need to be able to see how we can use metagenomic sequencing in trying to determine where we have resistance patterns by analyzing the DNA of these pathogens. And in that way, we'll be able to adequately put measures that address issues of infection prevention. With that, Lindsay and John, please feel free to add if there's anything I've left from my summary.
Yeah, I'll just add a little bit. Yeah, so basically what the genomics helps us do is it helps us to pinpoint where the problem might be, and it also helps us to investigate problems that may arise in the hospital. You know, for example, if we have outbreaks of resistant organisms, we can then try to pinpoint where this organism came from, how it's moving within the hospital, right? Where the transmission is occurring. I think before we're talking about transmission chains. So what genomics helps you to do is to figure out or to narrow down where in that transmission chain you may be having issues in your IPC, right? And then that also helps you address those issues. And it also helps you in antimicrobial stewardship, as in, because you will know what sort of antimicrobials your pathogens are resistant to, and be able to prescribe the correct antimicrobials so as not to further promote development of antimicrobial resistance by prescribing the wrong antibiotics. So yeah, so that's basically how I'd summarize it. Yes, thank you.
All right, thank you so much for that wonderful presentation. We really enjoyed it, and I think we have a few comments from the chat room. Tinta says, I've enjoyed the presentation. My question is, what sites, sorry, just try to see. Okay. What specific sites did they swab, and what guided them in choosing those sites? So that's the first question. Please take note of that. Then we also have someone who is asking, there's also asking if we can share the title of the study that was done. And then Sharon is asking, is looking at escape environmental. This is from Professor, sorry, just a minute. So, okay. Okay, so we have those two questions, and then the Prof is saying that there's someone who is looking at, I think, escape environmental pathogens at UTH for her MSc. Should be glad to get in touch. Okay, unless there's anything I've missed first from the chat room, maybe we can start with those questions. The first question on how the selection of the sampling sites was done. Thank you.
Sure. So standardizing environmental sampling collection is notoriously difficult. It depends very much on what your question is, as to how you go about the sort of methodologies of collecting those samples. For instance, if you have an outbreak of MRSA, there are sort of studies that say, well, actually using this type of swab picks it up better, or using sponge swabs picks them up better. So I think looking at the previous literature and understanding the question that you're actually, or the answer that you're trying to get from the question you're asking, is really, really important. In terms of how we went about it, we worked alongside the hospital estates team, and we did a sort of survey of the ward itself. So we had, the ward layout. We identified how many toilets and how many sinks and how many beds, sort of individual beds, and then the sort of mixed bays where you had multiple beds. And we went about sort of systematically sampling sites from all of these different areas. We knew on that ward that if the patient had been tested positive for one of these CREs, these carbapenemase resistant organisms, then they were isolated. So we knew that the isolation rooms were likely to potentially harbour more of these CREs in the environment. And in actual fact, generally they did harbour quite a few, but we found in particular one ward, one mixed use ward, bay was also sort of highly full of these CREs. But in terms of the sort of standardization, at the moment, there isn't really one standardized way of doing it. And that, as part of the work John mentioned, sort of in our future plans, is something that we're going to try and do, is to sort of develop this genomic tool to be sort of modular, so that you could, if for whatever use you potentially would want it for, you'll have the whole genome sequencing module, you'll have the metagenomics module, and you might, we're hoping to get it to the point where you're sort of saying, right, well, I'm looking at CREs, therefore this is the environmental sort of surveillance or sampling module that we would want to slot in place. If I'm looking at MRSA, well, actually, then this is what we would slot in place. But obviously with this sort of sampling, you have to kind of consider what it is you're looking for in the first place. There's no real agnostic way of looking at everything, although in theory you can, there's always biases that you have to consider when you're doing it. I hope that answers the question.
Thank you. I think it does. So I'll go ahead and read a few more questions from the chat room. So Kapula Chifunda says, sounds very interesting. Is there space for the private sector to be involved? And they're mentioning that they swab their surfaces monthly, and it would be great if they would have an opportunity to be able to apply this and be able to do some metagenomic sequencing. So that's the first. Okay, Dr. Sombo mentions eco-elevated, very cutting edge. Professor Mwansa will reach out to Sharon, and we want to know more. Okay, okay, and on the importance of implementing IPC measures, genomics is a way to go in AMS to fight MR. Okay. Then Mutinta mentions as well that as for hospitals that aren't able to use metagenomic sequencing, what manual methods can we use to track nosocomial infections in our critical care wards besides culturing? Please bear with me. The window I'm looking at is quite small, so when the message is quite long, it becomes a bit difficult to follow. So I think I'll end at those questions and be able to ask if we could have comments from the presenters.
Yes, I was going to talk about the one from Mutinta, where she was talking about culturing. So you know, culturing is like the gold standard. So there's always a need for culture, right? And you know, you can also use samples directly from clinical assays directly from the patient, and there are various PCR methods that are being brought up to be able to kind of genetically tell the similarity between those samples. So there are methods aside from whole genome sequencing that are cheaper. There's conventional PCR that's being brought onto line. You can use conventional PCR and then what we call RFLP, where you're digesting fragments of the DNA based on their sequence relatedness. And if you see a pattern, just like genetic fingerprinting, if you see that they have a common pattern between them, then you know that these isolates are more likely related, and you can kind of use that to kind of track the genetic relatedness of your sample and kind of track where your outbreak is coming from. As far as the private sector, yes, with our next project that we're looking to do in the NICU, we were also looking to do something within the private hospitals to see whether we can, be able to track infections using this whole genomic sequencing tool. And see if there's any difference between, our private hospital settings and our government hospital settings and, practices, IPC practices, and all of that sort of thing. So that we can learn from each sector to see how we can improve the IPC measures that are used. Basically, the treatment of our patients. So we get less nosocomial infections and less transmissions within our I hope that answers most of the questions that we received.
Yes.
sorry. I realized I forgot to answer the two secondary questions in terms of the sharing of papers. So the COVID study is already published. I don't know if we can share the link to that on here. And then, so the Oxford 48 is due to be published in Biomed Review by the end of this week, and then to be published properly. Well, we'll see how long that takes, but so they will be available. So if we can share those links. I should also potentially point people to we have a hub on the Global Health Network website. It's Pandora ID Net, and there's loads of tutorials and videos and resources and links on there regarding these projects, and also TB diagnostics. I can see John's putting all these links in, so thank you, John. So if you're interested in learning about how these things kind of work, and learning how Oxford Nanopore works, and learning about bioinformatics and things like that, then we specifically developed all of these resources to help laboratory scientists and researchers and healthcare workers. So yes, we can add that to your list of documents. So hopefully they'll be helpful.
Thank you so much for that wonderful presentation, and you can see the interest from the network in terms of going ahead and looking at it, going a bit at the level of metagenomic sequencing. So at this point, I'm going to invite our panel of experts in how we can be able to. Thank you, John. I think the link has been shared in the chat. Actually, it's Lindsay. Thank you. So I'm going to invite now the panel, our panel of experts to maybe mention one or two things that they would want to mention. Then we'll be able to end our echo session for today. Maybe I can start with Professor James Mwansa. Please, can unmute and go ahead.
Thank you very much. I had to switch off my computer at my low battery. But it has been a very interesting discussion, and it is quite clear that infection prevention is for everybody. And I think looking at the WHO competencies, and we have covered it very well, and it just emphasizes that we need to have IP programs in all facilities, and also emphasize that education and training should be continuous, and especially to look at the health-associated infections and continuous surveillance. I think that you've been very well covered. I did mention, I thought I wouldn't have time to say that let's involve the training very much. I have a student, Sharon Namkonda, in NICU at UTH, who is doing the escape organisms. And I'm sure I'll be very, very happy that the gentleman can get in touch with me as soon as possible. But otherwise, it has been a very good presentation.
Thank you very much, Prof. I'll now ask Innocent Hamuganyu to maybe mention one or two things from the two presentations. Okay, we can move on to Mrs. Musonda Mandona. I think we're able to see you.
Miss, and good afternoon. Please confirm that it's okay. Yes, thank you so much for the opportunity to contribute. I've been following the discussions closely, and I think the presenters have highlighted a lot of key points when it comes to IPC, the role of IPC and WASH. So really, without taking anything away, just to add that there is, I think, an opportunity we have in terms of linking IPC and WASH also with the biosecurity that we do and biosecurity measures that we can implement, not only in the lab, but also when it comes to agricultural settings, as well as when it comes to animals and animal husbandry. So I think that there is a rare opportunity that we have in implementing WASH and IPC effectively that can then prevent some of this, the emergence of not only resistant bacteria, but also just general disease that then stops us needing to use antibiotics, thereby reducing the inherent danger that we have with AMR. So I think just really emphasizing that it is truly a multi-sectoral approach, and there are other, I think, programs and principles that complement IPC and WASH, and using them together will ensure that we meet our goal of reducing this imminent threat of AMR. Thank you.
Thank you so much, and thank you for being available. Before I invite Alfonsina to give her comment, IT department, can we please be able to have, I think Dr. Sombo mentions, can we have a feedback form? Yeah, so with that, is Madam Alfonsina on the network? Okay, thank you so much. Again, to just appreciate all of you for being in attendance. It's been a very wonderful time that we've had. Let me start by thanking our presenters. That is Dr. Chileshe Lukwesa for the wonderful presentation, as well as Lizzie and John for those wonderful, insightful presentations. I want to appreciate as well our panel of experts, and further to appreciate the IT team who have been running up and about, helping us to ensure a smooth running of our ECHO today, as well as those that were involved in coordinating all the activities behind the scene, Madam Loveness, as well as the whole team from ZNPHI. Indeed, we really appreciate your efforts, and we look forward to when we'll be able to call you back, especially the presenters. I think John and Lindsay, I think there were a number of people part of the network that would probably want to have a further discussion and see how we can be able to engage, number one, the private sector. I think we are looking at how we can, we are starting to engage them in a number of these activities. And then as well as incorporating it into the training program, I think as Professor Mwansa did mention. So with that, I just want to say thank you to everyone, and our next ECHO will be on the 10th of July. That will be next month. So please join in again, and we can be able to enjoy this wonderful interchange of information. With that, we'll end our ECHO for today, and sincere apologies for going over time, and thank you so much.
Thank you.
Thank you. Goodbye.
Thank you. Bye-bye.
Thank you.
Thank you. Bye.
Thank you. Bye-bye.
Thank you. Bye.
Thank you.
Thank you.
Bye. Thank you, Mtinta. Appreciate